Good day everyone, I'm Sir Mavs and I'm back for today's lecture covering flowers, fruits, seeds, and embryology. But just a disclaimer, this will just be part one of two parts for the discussion on the reproductive parts of the flower because the flower alone, the discussion about the flower will be too complex. So I planned to make two parts for this lecture. So as an extension of the discussion about the plant shoot system, we will be focusing today's discussion on the derivatives of the shoot system, specifically the plant sexual reproductive organ. Let's begin. As previously elaborated from the past lectures, there were specific characters that define what a plant is. And one important facet is that plants were a product of long evolutionary history. Now, through this long process, one peculiar capability that plants were able to develop is the alternation of generation. The alternation of generation, sometimes called as metagenesis, is the alternation of a sexual phase and an asexual phase in the life cycle of an organism. The two phases, or generations, are often morphologically and sometimes chromosomally distinct. For plants, this alternation of generation is pronounced via the gametophyte and the sporophyte stages. The plant gametophyte stage is the sexual phase of plants wherein it produces sexual organs and gametes. Specifically for the sperm-producing structure, it is called as the antheridium, or in plural, antheridia. And for the egg-producing structure, it is called archegonium, or in plural form, archegonia. As we can all predict, the egg and the sperm will unite via the process of syngamy to form a zygote. Note that in the gametophyte or the gametophytic stage, the organism is in haploid or N. Now, once the egg and the sperm have successfully united, the zygote will develop as an embryo, thereby commencing the sporophyte or the sporophytic stage of the organism. The important thing about the sporophyte stage is that it is responsible for the creation of gametes. Note that in this stage, the plant is in diploid form. But some plants can be polyploid depending on their nature. The spore-producing generation further develops a sporangium and spore mother cell to commence haploid stage via meiosis. Once microspores are successfully created, it now again makes the process go all over again in a cycle. Again, from a haploid phase in the gametophytic stage, going towards a diploid phase in the sporophytic stage. That is the alternation of generation. The inflorescence. An inflorescence in a flowering plant is a cluster of flowers on a branch or a system of branches, or even sometimes occurring on stems. The inflorescence is categorized on the basis of the arrangement of flowers on a main axis or the peduncle, and by the timing of its flowering, may it be a determinate one or an indeterminate one. Through the evolutionary history of the flowering plants, or botanically termed as the angiosperms, many types and forms of inflorescences have evolved, and one of the most peculiar one is here shown. This is a typical inflorescence in the aroid or Araceae family. The aroid family includes some of the agriculturally important species like taro, gabi, etc., and other aesthetically or ornamentally valued ones like philodendron, monstera, and even amorphophallus species. Here shown is a spadic spathe inflorescence that is common in this family. The spathe is the leaf-like structure surrounding a central core called the spadix. Here. Interestingly, the spadix of Amorphophallus species are separated by some unique structures like 1. The appendix. The appendix heats up during full anthesis or flowering time. 2. An aggregation of the male and the female shown. Here shown in a tightly compacted cluster. 3. The peduncle. It serves as an attachment and support of the whole inflorescence as it remains attached to the base of the plant corm. There are other interesting inflorescences of angiosperms, and this is just one unique example. This is a close-up of the female flower of Anamorphophallus, and this is also a close-up of the male flower of Anamorphophallus. The flowering slides are examples of inflorescences. This slide shows a typical inflorescence of the sunflower family or Asteraceae, which is a head or a capitulum. The interesting thing about this inflorescence is that there are actually two kinds of flowers that makes up this inflorescence, the ray florets and the disc florets. The ray florets are ornated with flamboyant petals found on the periphery of the inflorescence, while the disc florets are aggregated in the middle portion of the inflorescence, typically turning black in most sunflowers during full anthesis. I would also want to elaborate the difference between a corymb, cyme, and umbel inflorescence. These three types of inflorescences may look the same at a glance, but differentiating them can be quite tricky, especially for the corymb and cyme, which literally looks the same. In terms of the arrangement of the flowers along the axis and the flowers creating a flat top, the timing of the flowering in a corymb differs from a cyme, such that cyme's oldest flowers are at the main axis, compared to the corymb, where the oldest flowers are on the periphery. Umbels are a little easier to differentiate, as the flowers emanate at a single point and the flowers diverge to it, forming a flat top. In terms of flower timing in an inflorescence, it can be further differentiated as either indeterminate or determinate. As for an indeterminate inflorescence, there is a point in time that flowers may temporarily cease to bloom as dictated by environmental factors. Moreover, the first flowers open up at the base of the inflorescence. Some examples include a spike, raceme, or even a corymb. Here are some examples of an indeterminate inflorescence. Comparing it with a determinate inflorescence, these have flowers opening at the top or at the middle portion. Moreover, flowers of determinate flowers are triggered to open all at once when specific conditions are met, example, during springtime. Some examples also include cymes, either a helicoid or a scorpioid, or even a dicacium, etc. Here are also some examples of inflorescences that we can find from different plants. Note on the complexity of branching, the form, and even the morphological attributes of the flowering inflorescences. Generally, for angiosperms, flower production is a phenomenon where plants really put an investment, especially in terms of energy requirement. Flowers are considered to be huge sinks for plants, most especially when in the long run it becomes a fruit. Flowers in most angiosperms are produced on active or even dormant buds through the production of a certain hormone called florigen. Again, that's florigen. Florigen is a plant hormone that triggers production of buds, specifically flowering buds. It is through the action of florigen that leaf bud creation ceases, thereby transforming the terminal portion of the shoot system into a highly structuralized reproductive organ. In this slide, we can evidently see the four major whorls of a flower, here labeled. That is, whorl 1 constitutes the sepals. The sepals acts for protection in the developing flowering bud, which may either persist during anthesis or until the flowering period, or caducus, meaning they shed off after the flowers have already reached full blooming. Whorl 2 are the petals, the main display of a flower that attracts pollinators through its irresistible and striking colors. Whorl 3 are the stamens, which consist the male reproductive structure, while whorl 4 are the carpels, the female reproductive structure. These whorls are arranged in layers such that the whorl 4 is the innermost structure and the whorl 1 is the outermost structure. When a certain flower fully matures and develops, the intricate structures found within this organ becomes more discernible and specialized, both in form and function. In this slide, you can clearly see the delineation among the whorls. Some important structures found on a flower are the pedicel, or the flower stalk. That is for an inflorescence, but if the flower is just solitary, there is only the peduncle. A receptacle is also present that is found on the base of the flower. In some species, this may be enlarged, forming false fruits, okay? Like that of your strawberry. That's a ripened receptacle. The petal, the stamen, which is composed of the anther and the filament, and the pistil, which is composed of the stigma, style, and the ovary. So this is a morphological representation of all the mature parts of your flower. In botanical jargons, there are some words that specifies a collection or aggregation of a typical structure or part of a plant. For example, petals are interchangeably used as applied using the word corolla, and sepals are sometimes called as calyx. Now, the aggregation of the calyx and the corolla is collectively called as the perianth. These botanical jargons may come in handy when you're dealing with a book or reading some botanical journals for references. On the other hand, an involucre refers to a collection of bracts that subtends the corolla. In this slide is a typical flower allied to the sunflowers, showing the overlapping sepals, here termed collectively referring to an involucre. A hypanthium, on the other hand, is a specialized structure that looks cup-like or tubular enlargement of the receptacle or of a flower. It is loosely surrounding the gynoecium or may even be united with it. In most flowers with inferior ovary meaning that the ovary is found as a distinct structure below the perianth. A hypanthium is most of the time used to refer to the swollen ovary. Moreover, a superior ovary is an ovary that is found, quote-unquote, raised above or within or the same level of the perianth. Further, flowers can be categorized as complete or incomplete, depending on the complexity in terms of the presence of the whorls. If the four whorls are present in a flower, it is deemed as complete, or botanically referred to as a monoecious. But if the four whorls are lacking, maybe one or two of the whorls are not found, okay, it is deemed as incomplete or dioecious. Thus, if all the whorls are present in a certain flower, it is at the same time denoted as perfect. This is a perfect flower. If any of the whorls are absent, it is denoted as imperfect, dioecious. Like for example, in this slide, there is a segregation of the male flower and then the female flower. In terms of the morphology of the flower, it can be further classified as either actinomorphic, sometimes called as regular or radially symmetrical flowers, or a zygomorphic, irregular or bilaterally symmetrical flowers. The main difference of the two is that actinomorphic flowers are flowers with more than one dividing plane, whereas a zygomorphic flower has only one dividing plane, which forms a mirror of the other. Examples: the sunflower is actinomorphic in nature, while an orchid flower is zygomorphic. Further scrutiny of the details of the flower. A floral diagram and a floral formula can be created. This is a floral diagram. This is a floral formula. The floral diagram gives us an idea of the number of structures found among the whorls and how they are arranged. In a similar manner, the floral formula infers a representation of the structure of a flower using numbers, letters, and various symbols, presenting significant information about the flower in a compact form. Note that in a floral formula, various shapes are designated per structure. That is, a triangular arc with a pointed tip represents a bract or a bracteole. For example, here for Hibiscus rosa sinensis. A dot represents the placement of the stem. Arcs represent a petal, shown in red color. A line with a circle on the tips represents one stamen, here. And the central circle represents the gynoecium or the carpel, etc. Once the floral diagram is correctly drawn, a floral formula can be depicted from this illustration. But in order to arrive in a correct flower formula, there are considerations that needs to be fully and carefully followed, especially in the assigning of the formula legends. Example: K, which denotes for the calyx, C for the corolla, A for the androecium or the male reproductive organ, G for the gynoecium, the female reproductive organ, and so on and so forth. Okay. Now, if we apply this and go back to the previous slide, okay, so there are other elements that are included in the floral formula, like BR and BL. Okay, so the first flower diagram has a flower formula of BRL with this cross pi, denoted by both the male and the female representation, with K, numeral number 5, C, 5, A infinity, and G, 5. Now, to interpret this floral formula, it means that there are bracts and bracteoles that are present in actinomorphic position in the flower itself. The female and male symbol denotes that the plant is complete and perfect. There are 5 calyces, K5. There are 5 corolla, but there is an arc connecting the corolla to the androecium, meaning that the 5 corolla are connected to the androecium. Okay? An arc denotes connection. The androecium is denoted by an infinity sign, but if you can count it, you have to put the number. Okay? It just happens that in this certain example of Hibiscus rosa sinensis, there are too many to count number of stamens, so it's just okay for you to denote it with an infinity sign. The gynoecium is superior. Why? Because it is denoted by a bar below G. Okay? If there's a bar below G, it means that the gynoecium is superior. But if the bar is above G, it means that the gynoecium is inferior. Okay? And there are 5 locules available. Okay? 5 partitions. 1, 2, 3, 4, and 5. That's how you interpret a floral formula. But of course, it is only when you were able to create a correct floral diagram that you can arrive to a correct floral formula. Moving towards the specialized structures, the stamen is the pollen-producing reproductive organ of a flower. Collectively, the stamens form the androecium. Zooming to the anther, pollen grains are contained within these sac-like structures. Then, the pollens mature and are ready to be released. Thus, the anther crack open via slits, sometimes called as the line of dehiscence, thereby releasing the pollen. Moreover, during the early development of the anther, there are tapetum cells encircling the microspore mother cells. The microspore mother cells will soon give rise to mature pollen grains. While the stamen, or while they are still maturing, these pollen grains receive ample amount of nutrition through the tapetum cells. During maturity, the microspore mother cells will now be giving rise to the pollen grains, and when the correct time comes, the pollen sacs will crack open via the slits to release now these pollen grains. Prior to the pollen being able to participate in the fertilization process, let's first recapitulate the important steps of its development. Pollen development can be summarized under two main processes, the microsporogenesis and microgametogenesis. Microsporogenesis happens before the production of a tetrad. It comprises the events which lead to the formation of the haploid unicellular microspores. During the process of microsporogenesis, the diploid sporogenous cells differentiates as microsporocytes, or commonly called as the pollen mother cell or meiocytes, which then divide through meiosis to form 4 haploid microspores. Each diploid meiocyte gives rise to a tetrad, here it is, of 4 haploid microspores, and microsporogenesis is complete with the formation of distinct single-celled haploid microspores. Then afterwards, the rest of the development will now proceed under the process of microgametogenesis, which comprises events which lead to the progressive development of the unicellular microspores into mature microgametophytes, which contains the gametes. This phase begins with the expansion of the microspore, which is commonly associated with the formation of a single large vacuole. The vacuole is very large, around 60 to 70% of the cell. Vacuolation is accompanied by the displacement of the microspore nucleus to an eccentric position against the microspore wall. Here. In this position, the nucleus undergoes first pollen mitosis, or the pollen mitosis 1, which results in the formation of 2 unequal cells. There is a large vegetative cell and a small generative cell, each containing a haploid nucleus. The generative cell subsequently detaches from the pollen grain wall and is engulfed by the vegetative cell, forming a unique cell within a cell structure. The engulfed generative cell divides once more by mitosis, which is the pollen mitosis 2, to form the 2 sperm cells completely enclosed within the vegetative cell cytoplasm, either before pollen is shed, or within the pollen tube. Tackling about the female organ, the pistil is the female reproductive organ of the flower, comprising of the stigma, which is the first receiving portion of the pollen, the style, and of course, the ovary. Across different plant families, the placentation, or the orientation of the attachment of the seeds via the number of locules, may vary. Here shown in the figure on the right is a lobelia plant placentation. There are only 2 locules, 1, 2, versus a campanuloid plant placentation, with 5 locules or 5 partitions. Okay. Ovules are the precursors to seeds, and as such, they are critical to plant propagation and food production. It's the place where the female reproductive cells are made and contained. It is what eventually develops into a seed after the fertilization process, only for the seed to then ripen and produce a complete adult plant. Ovules are contained in ovaries at the bottom of a vase-like structure, the carpel, which has a neck called a style and an opening at the top called a stigma. The different components of the ovules are as follows. The ovule is made up of the nucellus, the integuments, which forms the outermost layer, and the female gametophyte known as the embryo, which are found at the very center. The nucellus is the largest part of the ovule. It houses the embryo sac as well as nutritive tissue, and actually remains present in some flowering plants after fertilization as a source of nutrients for the embryo. The integument is a tough outer protective layer of the ovule. In the diagram here presented, we can see that the integument is almost enveloping the embryo sac. Commonly for angiosperms, like maples or daisies or other examples, typically they have a two-layer of integument. Okay? These two layers sometimes is referred to as bitegmic. Okay? Bitegmic or two integuments. The integument encloses the nucellus except for a very small gap, which is called the micropyle. Here. The funiculus is a stalk connecting an ovule or a seed with the placenta or the ovary wall. The calaza is the tissue where the integuments and the nucellus are joined. Nutrients from the plant travel through the vascular tissue in the funiculus and outer integument through the calaza into the nucellus. So this is a typical diagrammatic representation of an ovule and its different parts. In terms of the ovule development, it looks a little bit complicated, but every step of the development is actually very crucial. Okay, so we start from first here, one diploid embryo sac, or commonly called as the mother cell. This mother cell further divides into 4 haploid cells. Okay, but with these 4 haploid cells, only 3 will be degenerative, or 3 will die. Only 1 will proceed as a mature megaspore. Okay? This surviving cell will undergo growth through mitosis and becomes the female gametophyte or the embryo. The cells within the embryo further divides to give rise to specialized cells like the antipodal cells, situated on the top portion. Okay, so here, mitosis, further cell division from 2 going to 4, then it will now produce these different specialized cells. Okay, so let's try to elaborate the different specialized cells here. Okay. First, the antipodal cells, which are situated on the top portion. And in the middle are 2 polar nuclei cells, while on the bottom portion are 2 synergid cells. Okay, 1, 2. But at the middle of these 2 synergid cells is the ovum or the female gamete. Okay, again, antipodal cells, polar nuclei, 2 synergid cells. In the middle, there is the ovum or the female gamete. Later in the stage, only 7 specialized cells remain in the embryo sac. This consists of 6 haploid cells. So all of these cells, the top portion, the bottom portion are all haploid, while the cell that is found on the center is a diploid one. Okay. Now the reason behind will be fully elaborated through the next slide. because it will now be acting or participating in the overall process of double fertilization. Okay? So plants do undergo fertilization, but it is in a double manner. Okay? That is, in a single pollen grain that attaches to the stigma, and of course, with proper recognition, no, hindi po pwede na ibang pollen mag-fertilize sa ibang plant. It is only species-specific or infra-species level. Infra-species meaning these are species that is lower than the classification of a species level. Okay? So this may be the varieties, subspecies, forma. Okay? But the point is when a certain pollen grain is already recognized by the stigma, it will proceed downwards and accepted by the pollen tube. Now, if a certain plant pollen that is of a different species enters successfully in the pollen tube will have specific enzymes within it that will secrete digestive juices or something abortative in nature. Okay? So that really determines is high specificity species level fertilization for your plants. Now, assuming there is a successful fertilization that happened after the pollen grain was successfully permitted by the pollen tube, it will produce two different sperm cells. Okay? One sperm cell will proceed to fertilize the egg, forming the diploid zygote. Okay. The cat is noisy here in the campus. Okay. Here, two sperm cells. One sperm cell will enter and fuse or fertilize the egg, forming the diploid zygote. Okay? Where is the egg? Siyempre, hanapin niya ito. Diba? Magkakamukha yung mga cells na tatlo dito, pero sa gitna lang pipiliin niya kasi andun lang yung egg. Yung isang sperm cell, i-fertilize yung egg. While the other sperm cell will fertilize the two polar nuclei to form the triploid endosperm. So, N plus 2N, 3N, triploid. Okay? Galing, no? This triploid endosperm will soon cater for the nutritional needs of the growing embryo. Okay? So, again, a recap. A pollen grain successfully lands through the surface of the stigma. The stigma permits the pollen grain to enter through the pollen tube. The pollen tube recognizes the pollen grain. It further enters through this long canal. Then the pollen grain will emit two sperm cells. One sperm cell will fertilize the egg, forming the future developing plant, while the other sperm cell will directly fertilize the two polar nuclei, forming triploid structure known as your endosperm. The endosperm will soon facilitate as a nutritive source for your developing plant. Okay? Now, the remaining portion of this chapter, specifically for the fruit, seeds, and the embryology of plants, will be discussed in the part